Let me unmute myself. Okay, thank you so much for that introduction. It is my pleasure to talk to you all here today, and thank you for taking some time to come and really think critically about how you're managing your research data and ways that you can make it better. And I agree that managing data and research data itself is a beast, and I really appreciated that the DALL-E mini AI, when I put in data lifecycle, it came out with this sort of abstract, strange art piece that I think really encapsulates how nebulous research data can be. So let's get into what we're going to do today. So my main objective for you today is to take you from feeling like this to feeling a little bit more like this, which I love this stock photo because it's got all of their research data neatly organized on the bookshelf. And of course, who hasn't sat at their bench with their microscope and their beakers of different colored liquids? So this is definitely, a true portrayal of the ideal for science. But, more seriously, I have three main objectives for us today. One is going to be to go over the concepts of data management. Why is managing research data so important? And really, it gets to the meat of enhancing rigor and reproducibility of scientific research. And then I'm going to give you some tips and lots of resources so that you can continue to dig into this further to really upgrade your data management practices. And hopefully in the long run, that's going to save you time. And then also to bring it all together and establish a data management plan for your lab or for each project to help with cohesion and really reducing delays when transferring a project from one person to another. And you heard about this a little bit in the introduction, but I really want to stress to you that I've been there. I've worked in research labs for many years. I've done both basic bench science, and then I've also done clinical work and clinical research and field research on human subjects and in global health. So I've been in lots of different research environments and kind of benefited from good data management practices, but also felt the difficulties and sting of when data isn't managed properly. And then I also want to make sure that we kind of are all on the same page as to what falls under this umbrella term of research data management. Maybe when you're thinking about data management, what comes to mind is what to do when you collect data and process and analyze it. But that's really only one section of the data lifecycle. We want to start thinking about how to manage data in the planning stages when the project is just in its infancy, thinking about really documenting well protocols and workflows, and then taking it through that data collection and analysis steps. And then also on the flip side, what to do with data once it's been collected and you've reported it. How do you save it for the long term and for future generations or for yourself in the future so that you or others can use it again? And so I'm going to talk about tips and different things you can do at each of these different stages. And all of that then falls under a data management plan or a data management and sharing plan. It goes by a couple of different names. I may also refer to it as a DMP. Okay, so I would really like to start this off with a bit of a personal story of my own experiences. I worked in a parasitology lab that really excelled at research data management. So this was the parasitic worm that we worked on. We were based in St. Louis, and we worked with groups all around the world. And what we had was that the group had been around for many years. And so we had about 30 years of a biobank of samples, but also the clinical data that went along with that. And what that allowed me to do when I came new to the lab was use these old samples and leverage the data that we had about them to learn new things about the parasites that we were working on. And so it didn't rely on new collection. So that really also increased productivity because there's less time collecting samples. It also makes you more competitive for grant funding if you can reuse data, especially in these types of collection projects, because like the NIH or other funding agencies are really reticent to fund collection-only types of projects. And the fact that our group was good at managing data and managing projects made us good collaborators. We were easy to work with, and that was how we were able to, one of the ways that we were really able to expand the research across the globe. And so those were all really intrinsic, internal motivators to practicing good research data management. But if that doesn't convince you to do data management out of the goodness of your heart, there's also many external motivators that'll help get those wheels turning. So we're really in the middle of a culture change in scientific research that's pushing for more open data, more open sharing of the products of research. And in response to that, many journals now mandate the depositing of raw data to a public repository. So you used to be able to do, let's say, a proteomics project, and the raw data was not necessarily associated with that publication, only the aggregate form. And now, if you go to submit a paper with proteomics data in it, you will be required to submit that data to a public repository or have it shared in a certain way before you can even hit submit on that manuscript. So knowing about repositories and how to manage and properly format that data prior to submitting to a journal is critical. And then also getting your research funded is also going to require data management plans. So the NSF currently requires a data management plan for grant proposals, and the NIH will in 2023, next year. So I'll talk about that in a little bit more detail. But all of these groups really stress that the research outputs should be so-called fair, and that stands for findable, accessible, interoperable, and reusable. So the data from publications, you should be able to find it and access it and download it and understand what it's saying and use it again. And not only reusable and interoperable by people, but also by computer software. And again, these slides are going to be made available afterwards, and I have lots of links, usually in the corner, where you can learn more about the particular practices that I'm talking about. And so, why is this so important? So, you can see this table here. If someone wanted to share this data set, they would really have a problem with it. It's really not cleaned up. And so there's lots of inconsistencies with the date, and there's empty fields, and there's actually multiple data in each cell and things like that, which really limit its reuse. And even if this is your own research, if you came back to this file 6 months from now, would you remember what all of these different acronyms meant? Maybe, maybe not. And so one of the things that's important with data management is making sure that the files that you have are documented well enough that you or someone else could come back to them years later and still understand what they mean and that they don't lose their value. So I want to stress to you that good data management is not just something that feel like you're being forced to do, but to think of it from a mindset that it's a love letter to your future self, because the person who's mostly going to benefit from these good practices that you're going to do is you. And that's really well shown by this little cartoon where someone asks, how's it going with your data analysis? Oh, I can't understand. And they say, who collected the data? Oh, I did 3 years ago. And I think any of us who have worked in research before can definitely recognize this kind of pain where you didn't end up documenting the right things that you needed months or years down the line. Okay. So that's sort of the why we should practice good data management. And now I'm going to go into some specifics of how you can do this across the data lifecycle. So again, we're going to start here in the planning stages and talk a little bit about protocols and workflows. So you probably know what a protocol and a workflow is, but I just want to be really clear that these sequences of steps in a process applies to both physical processing and computational. And it's both before data collection and after. So we think about having a protocol when we maybe grow up some cell line and lyse the cells and collect protein, and then we do something with it. But it's also, once you have the raw data, let's say you used a plate reader and read a 96-well plate and you got the raw data, it's also what did you do to transform that data to the final figure that's in the publication. And I know that I've definitely been there where I have a graph that I thought was really interesting, and then I can't remember exactly how I got to it. And so then I have to reanalyze the data again so that I can make sure that like the method section of my paper is correct. And so by documenting all those steps, you know, save yourself some time. So some better practices in regards to protocols and workflows is to really maintain a standard set of workflows and protocols and keep old versions. So, you know, come up with a labeling system for the different versions, and then that way you can note versions each time you use it. This is super important because protocols tend to change over time, and by the time you go to write up a paper, the version of the protocol that's available now might be slightly different from when you did the actual experiment a year ago. You also want to make sure to keep documentation of how data is transformed, and especially now that things can be very complicated, keeping documentation of computer scripts that was used to transform data. And then another one that you may not be as familiar with is these RRID numbers. These are Research Resource Identification Numbers. And this is really a unique identification system that's maintained by the NIH that allows you to track molecular reagents. And this evolved because, you know, let's say in a method section of a paper, you would say, I use some mouse monoclonal antibody to beta-actin. And then the traditional way of saying which one you used would be to put the company and the location of that company. And this is really an archaic way of doing things because, as you know, if you went to Santa Cruz, you can imagine putting in beta-actin is going to return a lot of results. And then you're still left with the question of which of these is the right one, because, you know, not all antibodies are the same. And so the RID numbers is a unique identification number for each molecular reagent, like an antibody or a cell line. And so if you put in that catalog number for the beta-actin antibody that you used, it'll spit out all of this highlighted information, and you can include all of that or some of that in your method section so that the person reading the paper knows exactly what you've used. And then also in your lab management, you could have a file, like a spreadsheet of just all of the molecular reagents that you use and their corresponding RID. And this is also useful if your group is developing molecular reagents. You can apply for an RID number, and it'll allow you to track the antibody that you made and who's using it and how they're using it, or the cell line that you created, things like that. So here's an example workflow with integrated data management. And the workflow itself is in black text, and the integrated RDM is in red. And so, for example, let's say we have a 96-well plate that we're growing some human cells on. We infect them with a virus, and we're also adding an inhibitor to the virus. So before we even start, we would want to make sure to have like a unique ID for this whole set of experiments that's going to go along with this, and make sure that there's a complete detailed protocol with the version, and then some information maybe on some of the reagents that you're using. And then let's say we're monitoring viability, you're going to get raw numbers from a plate reader. So you keep the raw data, you also keep the aggregate data, and you'll have a workflow that describes how that data was transformed. Now, for something as simple as this, where we're just getting a mean and some error bars, it's like not that involved. You probably could repeat that very easily, but for some more complicated types of graphs, like for example, for this one, you may want to know exactly how the data was transformed from this image of showing every green cell is a viral infected cell. How did we get from an image like this all the way to the aggregate analyzed data? And then also, I want you to pay a specific attention to image files, because You want to make sure that you've got a designated location and good labeling for all your raw images. Knowing the provenance or the origin of cropped files, and clearly designating which of each of these files is used for the representative blot. Because if you over and over again do this same experiment, they're going to all start looking the same. And you want to make sure you know which is the one that's in the paper, which ones aren't in the paper, but are other good images. And then when it comes to publishing methodologies, there's some real systemic weaknesses in the publishing field. And this all really stemmed from the word count limit that was there, and which we've really gotten over now, right? Because, journal articles still often have word count limits, but because things are available online, you can always add in supplemental materials or other files so that you can have a more extensive methodology presented with the work that you've done. And this is an example of the more traditional methodology publication. This is from one of my papers, so I've done this too. But QPCR is such a common run-of-the-mill technique nowadays that we really don't explain it very well in the paper. We say it was done as previously described, and this really lacks the necessary detail to be reproduced. And then typically you end up clicking on these links and it just leads down to a reference rabbit hole without any answers. So, when publishing, you want to consider publishing the methods alongside the paper. Lots of journals have will suggest that you can do this, like the Cell journals have STAR methods, there's protocols.io. So, making those extensive methods available with the publication, making scripts available. And then if you've got some new method, you may want to consider publishing a report in a methodology journal. And just here's some more links again about workflows and protocols. So now I'm going to move on to file management, and in this case, file mismanagement in this, in this comic here, where their documents folder is completely unreadable, really, and you wouldn't know what any of these documents were unless you opened them all up. So, my rule of thumb is if it takes you more than 10 minutes to find a file, you probably have a file management problem. And so, what I encourage everyone to do is come up with a file management system that works for you. So, for example, I've got two here. One person has organized by file type. The other is organized by analysis type. I fall into Camp B. I like to organize my files that way. But if that doesn't sit well with you, figure out a way that works with you and do that. I think for person B, that fits better with if you have to do a lot of pre-planning before you do a project and you can kind of envision what all the figures are going to be. And so you can sort of set it up that way versus this file type version is maybe more for a sort of an exploratory type of study. And I would encourage you, if you do organize by file type, that once you get ready to write up a manuscript, to make like a production folder where you take everything out of those file type folders that are involved with that publication and put them all in, maybe just a copy of them into a production folder so that you can keep everything for that one publication together. Same thing goes for grants. So come up with a management system and implement that. And then I also want to talk about some tips for better file naming. This is both for files and also for like column headers in a table. So use a standardized date. Year, month, date works great because it eliminates the ambiguity between the US versus European dating system. Make sure you're organizing by project, again, if that works for you, but especially having unique identifiers for different projects. Include versioning, especially on things that you're writing lots of drafts for and revising, like grants and manuscripts. Try to use underscores, but not spaces or periods or special characters. This table here shows some of those examples. Computer software has trouble with this column here, some of the characters in this column. And in the end, just being consistent so that you know what's in a file before you open it based on the name of the file. And again, here's some more tips. Okay, so next I'm going to move on to documentation. Documentation is so important, and it really encompasses everything with research data management. And so, you'll have to think about what way are you doing it now? Are you using a physical notebook? Are you using an electronic notebook? Do you have a hybrid of the two? What works for you and makes the most sense for the type of research that you're doing? I personally used more of a hybrid version where I had a physical notebook, but I also had copies electronically, because the pros of an electronic notebook is so much easier to reorganize things, to keep parts of a project together, and also to be searchable by keywords. But no matter what format you use, you want to make sure to keep or link all data for one project together. And again, we're going to go back to the ideas of protocols and workflows, reference those by version, and note divergences. And if you have a good handle on your protocols, then you can easily say, hey, I'm using Western Blot protocol version X, and I changed this and this and this. And so you're not really documenting everything about that protocol, but just the key changes. And then again, integral to documentation is maintaining good file management. It's important to keep electronic logs of physical data and physical samples, and then including a README file, which I will talk about in a little bit. I did want to take a second to let you know that UAB supports, we have a subscription to the Lab Archives electronic research notebook. There's a link to log into Lab Archives here. It's free for you to use because UAB pays for it. And there's lots of training online, and there's also, we also schedule training throughout the year.
I actually have a question.
Yeah, can you put the question in the chat?
Sure, I can put a question in the chat.
Yes, we'll get to your question in just a moment. Okay. So in terms of documentation, I do want to take a second to just make sure we're all clear on the difference between data and metadata. So data is the outputs or observations from any systematic investigation. So for this example, we've got a picture of a cat. That's the data. And the metadata is structured information about that data. So in an image file, if you right click and you can see the image info, that's the metadata about that image. So it'll tell you the file name, the file format, maybe who took the picture, when it was taken, maybe some geolocation information. And so metadata is really important because it helps you stay organized and it can help you find things. So for example, why do you need metadata? Why do you need to keep track of that? So for example, here is a pretty simple table from one of the studies that I did. And if I just gave you this table, you probably would have some trouble interpreting it. Let's look at this date here. 18 were registered, 16 attended, retention was 89. I'm guessing that that's percent, but it's kind of hard to tell. Metadata gives you more information about those column headers. So having information about each of those columns, what type of data information they are, and then a little description about those. And so then now this table becomes more interpretable. So if I was sending this to a collaborator and I just sent them the data file, that's not going to be very helpful. But if I also send a little table about the metadata, then it becomes more useful. And this is really important to think about when you're sharing data in a repository, what types of metadata are going to be necessary for someone novel to the project to be able to interpret those findings. The other thing that I love to use metadata for is upgrading your table of contents. So this is a screenshot of a part of one of my table of contents. And so what I would do is for each experiment that I did, I got a unique ID number or code. I would write a very short description of what was going on in that project, and then some keywords into the methodology, inhibitor, what system was being used, etc. And so that way this table was searchable and I could find all of the experiments where I used a certain virus or all the experiments that belong to a certain publication or maybe a grant submission. This is a really important column to have because as we all know, many of the experiments we do don't end up in a publication, but it can be really important to try to find the ones that are involved. And this was great for me when I was in the lab because I could search this and keep an idea of where things were if I needed to go back and look at something, but also really invaluable to my PI when I left the lab that I could give it to them and they could, you know, find important things quickly. So now I'm going to talk about the README files. So README files are really, they're going to be a time saver and stop you from having to click through everything in a folder. So let's say you started a project 6 months ago, it kind of got on the back burner and now you're coming back to it and you've got this whole file folder of a bunch of random stuff. Or maybe you're passing a project on to someone new. Instead of going down the rabbit hole of opening up each of these subfolders and seeing what is going on with this project, it's nice to include a README file in the parent folder that summarizes the project and the files within. So it's just a text document. I recommend that you make a template and you're not going to write this up, type this up new every time, but you'll just be copying and pasting important key elements from maybe different files or different file names into this document. That's going to give you a little summary of the project, maybe where you're at in the project, what data types are there, what files are, what are each of these files, what are they, what's their metadata, maybe spelling out some acronyms that you don't routinely use, what protocols have been used. One, I think that's super important is where are the samples located that are, you know, relevant to this project, because it can be really challenging to match things in a lab notebook to the freezer, what's in the freezer. And there's some more tips, extensive tips to writing these README files. So I'll give you another example from another personal example where we had a breakdown of file management and documentation. So the parasitology lab that I worked at was great with clinical data, but we were new to the proteomics field and the group made an error. That was actually really costly in terms of time. So what had happened was that, you know, over the years, we had accumulated over 70 different sets of experimental preparations of either from a patient sample or from a specific parasite that had been prepared in a manner of ways. And then those were sent to the proteomics core facility where they determined what proteins were present in the sample. And then they sent us back these LCMS files that told us about the proteins and peptides that were in the sample. Well, unfortunately, no one had set up a system where we linked the prep from the lab notebook to the data file. And so when it came time to write the paper, we really didn't know what went with what. And it took several people, several months to match these back up. And this could have been so easily avoided in the forefront or in the onset if they had just established a common language between the two research groups and had a key for the data sets. So now how they do it is that they have a study ID number that's a unique ID number for each experiment. And then it gives you information about the sample preparation, and then it links it to that LCMS protein file at the end. Okay, so moving on to storage. Really, you need to think about not storing data or research material on your personal computer. This is not only that's not only a bad practice in terms of research data management, because it can get lost, or if it gets stolen or your computer dies, you lose the data, but it's UAB policy to not do that. So we have lots of other safer options, Box, or you may have a departmental server that you're able to use. And what's important about these is that the files are backed up right on a regular basis, and old versions are archived. And then the other thing you need to think about with storage is complying with the security necessary for PHI. And so when you're working with any kind of sensitive health information or just sensitive information in general, you need to follow your IRB protocol. And de-identify the data accordingly depending on how you're sharing it. So this table just kind of talks about that you need to de-identify further and further for maybe a public repository access versus when you and your collaborators are using it. But all of this should be spelled out in your IRB, and you can also talk to UAB IT if you have some questions about how to make sure that the data is securely behind, safe. And then once the data has been collected and you're getting ready to publish, or you've already published, you need to start thinking about long-term preservation of the data. And the best way to really do this, I think, is to take it out of your hands completely and choose a repository to maintain the data. It's going to maintain data integrity, and it's also going to increase data reuse and also citation. So if you're just keeping it here at UAB and nobody can access it, then you're not really opening yourself up to being able to share the data and maybe find some new collaborations or new discoveries about that research. The rule of thumb is to try to find a more subject-specific repository if you can, if it's appropriate for the data types that you're interested in sharing. But if not, and there's just some examples here, but there are many out there. And then if not, you can use a generalist repository. Many of these are also free to submit data to. One of the things that I offer is a consultation where we can talk about what data repository would be the best fit for your data type. And I just want to give you a little reality check. You probably already know this, but if in your paper you write, "We'll share data upon request," then yes, technically you are complying with maybe some of the journal's data sharing policy. But in reality, this does not actually amount to data sharing. So this is an article from Nature a couple of months ago, and the authors requested data from 1,800 or so manuscripts where the authors said they were going to share. But what you can see here is that of this number of manuscripts, almost all of them either did not respond or they declined the request for data. And then only 120, which is 7% of the authors actually handed over their data. If you put your data in a repository, then you don't have to worry about being emailed about this, and you also don't have to worry about finding the data later. It's just already there, and somebody else can download it. And then it also, when you submit your data to a repository, it's given its own unique identifier, like a DOI, and they can cite that. So they'll cite your paper, and they'll also cite the data set. So you'll be getting more citations and getting the proper regard for your work. And then, I also wanted to share this story, that really bad things can happen without a dedicated repository. This was a huge undertaking, the Saccharomyces yeast deletion project. This was done in like the late 90s, early 2000s, and this was over 16 labs that looked at deleting every gene from yeast as a model organism and seeing, if they could determine the function of each of these genes, what they do. And they have lots of publications about it, and we found a lot of really cool information. However, they also had a link to this website that was hosted by Stanford, and you could go in and you could get information about specific primers and things like that. And that link is now dead. So at the time, there really wasn't a lot of dedicated repositories for data, and so the website was the next best thing for them. But nowadays, you don't have to do that. You can use a repository. It will have a DOI, so you don't have to worry about link rot, and your data will always be available. Okay, so moving on to the last part of the session, I want to talk about how we can put this all together and take all the data management practices that you're doing or you're going to implement into your group and put that into a data management plan. So a data management or data management sharing plan outlines what type of data is going to be collected, how it's going to be analyzed and stored and shared. So in our little cat analogy, the data management plan might say something about how the files will be stored, that they're going to be analyzed with this specific software. How are they going to be stored? How are they going to be made available? And something about maybe the software code that was used. And this is so important right now because starting in really the next grant cycle, NIH is requiring a data management and sharing plan for all research grants that are dealing with data. So they really have two main points. One is that there's a submission of a 2-page plan, and 2, that in your annual RPPRs that you'll document compliance with the approved plan. So that 2-page plan has 6 different sections in it: description of data, tools and software, data standards, how data will be preserved and when, concerns around distribution and reuse, really safety and security, and then who's going to oversee the management plan. So I'll go into those in a little bit more detail in a second, but I think, you know, what's probably in the forefront of your mind is, okay, does this apply to my grants? So starting again, January 25th, it'll be all research funded by the NIH that generates scientific data, including extramural grants and contracts, intramural grants, all NIH funding mechanisms. And they just released what project codes are going to fall under this category. So it's R's, most K's, SBIRs, STTRs, and RC grants. Now the policy does not apply to funding for things that don't generate data. So training grants and fellowships are exempt, conference grants, all these ones in the red parts. So if this does apply to your grant, then you know you have to write that 2-page plan, and you're probably now wondering, okay, how is this going to affect my submission? So the plan is submitted with the proposal, and with that you can request funds for data management and sharing costs. But the good thing for you now is that it's not going to be reviewed by the study section, and it does not impact scoring. So it's going to be reviewed by NIH staff just to make sure that everything you've put in there is feasible. And if there are things in there that don't, that maybe aren't going to work, then they'll contact you and help you revise the plan. So the plan itself is not set in stone. It can be updated as the project evolves, just like you're not, you know, locked into doing every experiment that you propose. You're also not locked into sharing the data the way that you outlined in the beginning. So you can update that plan. And it's important to update it because non-compliance with the plan could impact future funding decisions for the recipient institution and yourself. And this is a great website, sharing.nih.gov, that has details on all of these new guidelines and the new policy, and lots of information about, you know, as they're continuing to develop more information about this and clarify how the policy is going to work. Okay, so not everything falls under the data sharing plan. You're not going to be required to like share your laboratory notebooks. Preliminary and optimization types of experiments do not fall under this category. You would not be required to share like the raw case report forms, and this does not extend to any physical objects or specimens. And you're also not expected to share any kind of correspondence with colleagues or drafts of paper. None of that is part of the data share. But they're actually fairly clear on what data should be shared. The older policies from 2003 were not as forthcoming with this information. So the policy does define scientific data as the recorded factual material commonly accepted in the scientific community as of sufficient quality to validate and replicate research findings, regardless of whether the data are used to support scholarly publications. So what this means is that you need to share, or you need to at least explain whether or not you're going to share the underlying data from published studies, but also data from a study, even if it doesn't end up in a publication, and any null findings, again, that don't result in a publication. So those last two are a little bit newer. And the policy also stresses for you to share, as much as possible. So be of the mindset that I'm going to share this unless XYZ. So, but there are acceptable reasons to not share, and writing in the plan, you'll just explain why you can't share. And I've organized these from strong to weak. So of course, if there are any laws or regulations that prohibit disclosure, then you'll follow those laws. This does not trump the laws. And then we want to be very careful when any concerns about privacy or safety of research participants. Those could be very good reasons to not share raw data. And then especially now as we're transitioning into the new policy, if you're working with data sets, you need to think about their consent form for participants and whether or not that consent form included the ability to share certain types of data publicly. And if it didn't, then you're not allowed to share that data. And then moving forward, you want to think about writing consent forms in such a way that it will allow you to share maybe certain levels of data. And then the last one, which is just wording from the NIH, which I don't quite understand, what this means, but it seems like a pretty weak reason not to share, that the digitization is impractical. So I can't think of an example of that, but as this policy continues to become implemented, maybe we'll get a little more clarity on what that exactly entails. Okay, so now we're going to talk about the 6 different elements of the data management plan. One is the description of the data. So what types of data will be collected and used? Are you doing CT scans? Are you doing microscopy, sequencing? And then what level are you going to share? Are you going to share the raw data files? Maybe, maybe not. Are you going to share aggregated data files? Talk about that. And then what types of metadata will be included? You need to explain any software or code or tools that will be needed to access the shared data. So you don't need to talk about every piece of software that you're going to be using to analyze the data throughout the research, but you do need to talk about ones like if you're using SPSS, will someone need SPSS to access the data, or will you be exporting it as like a CSV file, and so you don't need to worry about the SPSS. And then making sure that the tool if there is a specialized tool that's being used, that it's not going to be unavailable when you still need to access the data. And then data standards, thinking about formats of data and data dictionaries and common data elements. These are all things to think about so that the data itself is interoperable and reusable by someone else. So I've just given an example here of a very untidy data set where things are not all in the same format. And so that would be a problem for someone trying to reuse that data. And then if there are no consensus standards, then you can just indicate that the type of research you're doing doesn't have any standards. And so these are the standards that you're going to come up with and use. And then in the 4th section, you want to talk about where the data will be shared and when. So what's the repository and how are the data going to be identified from that repository? And the data must be shared no later than the time of publication or the end of the performance period of the grant. So if your grant is 4 years long and you still haven't published the data, you'll still have to, or published a manuscript about the data, you'll still have to make the data available. In the 5th section, this is really dealing with concerns about distribution and reuse. Are there any limitations to the sharing and why? You just need to explain and what protections are in place to make sure that the data, if there is any sort of sensitive information, that's going to be maintained and safe. And then the last section is who's responsible for the oversight. Most of the time, this is going to be the PI of the grant, but it doesn't have to be. And just talking about how they're going to make sure that everybody in the group knows about the data management plan and how they're going to make sure that everyone is complying. And we have, me and some other librarians have worked on building out a directory of over 100 example data management plans. They're not necessarily the new format. In fact, most of them are not. But you can also ask me if you want a sample of the new data management plan format. And we've also got templates and other things like that. So, in closing, I want to let you know that this is a lot, and it could be very intimidating to try to implement this all at once. But I think there are a few small things that you could do right now that would really up your data management game. Start standardizing file naming going forward. Make sure you're versioning things. Keep projects organized. Start including README files. Make sure that you're storing data electronically with automatic backups. You really already should be doing that. Again, CYB policy. And then for each project, devising a plan. Talk to everyone in the research group about how we're going to make sure that we're documenting things correctly, storing things correctly, and sharing that data. And making sure everyone's bought into the plan. And before I take questions from you here, I just want to let you know that you can reach out to me at any time. We have a website, we have a Twitter feed, and I'm available to give data interviews, to assist in prep of data management plans, help you find a repository, talk to you about lab notebooks, and give consultation instruction on any of these data management practices that I talked about today. So talk about those in more detail. And just really quickly, there's some additional resources that, again, you'll get in this, and there's some upcoming UAB library programming that's you should check out and sign up for. And with that, I will stop here and take any questions that we have.
Thank you, Marla, so much for that. Very, very informative and very useful. We have a number of questions. If you want to keep continuing putting questions in the chat, some of these have already been answered. So about lab archives and stuff and resources. Let's pursue a little bit this. I think all of us who are writing grants are going to need to know what these things should look like. So, I mean, 2 pages from one point of view seems a lot. On the other hand, from what you're saying, 2 pages is nowhere near enough. So can you talk about that a little bit?
Yeah, absolutely. And that's part of the reason why we put together this directory of example plans. One thing that I did is I took a resource sharing plan, which, those in the grants, they're probably about half a page. And that kind of, you could take that and sort of morph that into a data management and sharing plan. So I've done that. And if anybody wants that sample, I'd be happy to share it with you. But it's like the first section that's about what data should be included. It's really about data types. It's not every single bit of data that you're collecting. You don't want to rewrite the whole proposal in the data management plan, but you may say, I'm collecting clinical metadata, I'm doing proteomics, and I'm doing metabolomics. So those are your 3 data types. And maybe for the clinical data, you're going to share that as aggregate, and you'll explain what level. And then for the other 2.
Let's talk about a little bit more prosaic. Those are big data sets. And I suspect that's not quite such a problem, because people who do metabolomics, proteomics, etc. are used to this. What about you doing Western blot? What are you supposed to put for that?
So there are standards out there for that. And you may say, I'm going to put the raw images all right now it's like I have representative blot and you just kind of stand behind that. But there are options to share your other replicates, maybe either in a supplemental file, or you can put it in a data repository. And so that would be more rigorous, right, to keep that. So you could mention something about that. And you don't have to talk about every single experiment that's ever going to happen, because it's impossible to know what those are all going to be.
Yeah, but not the individual experiment, but for example, in many things, the actual level of technology is fairly simple. Like but I think actually having something which is the actual current protocol with examples of not of high level data, but of a very simple grant where you're doing animal knockouts and Western blotting or PCR or that sort of thing will help people.
Yeah, absolutely.
yes, people are, Matt is answering all your questions for you here, but he's answering, he seems to be answering them correctly, so it's all fine.
Yeah.
And yeah, so Peter, you had a number of questions about, and I think you raised an interesting point. So there's this position, where is Peter? He's on the call somewhere, but there is this position that, which is the case, that UAB owns the data we generate as institutions. So are we really free to go and deposit it all over the place, or does the institution have to go and do that? And what does it mean, even ownership of data, if we have to give it to everybody whenever?
I don't know if I can answer those questions, but I mean, they are good questions. Yeah.
Yeah, I'm not sure what that really means when it comes down to it.
Yeah, I mean, in the end, the ownership is like, if the data is publicly available, it doesn't mean that I can just use it and not give credit where credit's due. So the ownership is still there, and I'll say, hey, I used Peter's data, and now and I cite him in my publication.
Right.
So it's available, but it's, it's not yours.
I guess I was thinking of sort of unanticipated commercial value of
Sure.
the stuff that you deposit, right? I did a
Yeah.
huge screen. I wasn't really looking for this, but buried in there is something
Right.
of real value.
Yeah. Well, yeah, I don't know.
Yeah, it's an interesting. There are a few things there that are not being, and NIH has its own proprietary position about IP generated through its own projects, actually. So if you put it out in the public domain, as everybody knows, it's no longer suitable for IP. You've disclosed it. Interesting. Well, thank you, Marla. That was a really brilliant exposition there of these issues. I feel very grateful that you could spend your time and do it. The thing will be recorded. We will distribute it widely. And I think as we get closer to this deadline, there's going to be a lot of panic. So I think in the next thing we're going to ask you to do, I think, is to sort of go through what does this example actually look like, and what would you do to put things into it?
Yep.
Well,
yeah. And I am giving a talk solely on the 2023 policy in November. So keep an eye out for that.
Well, if you just make sure we get it, we'll make sure everybody on this call and the whole other people who've been an interest get it too, right?
Yep. Okay, great. Well, thank you, everyone.
Thank you. Bye-bye, everyone. If
Thanks so much.
Stay on a second, Marla.
I will.
I know why all these people are still there.